So now we're going on to our graduate student scholarship awardees presentations. And they are all PhD candidates, and I will list their names and they will present one after the other. So we have Gao Jinghu from the Department of Biochemistry, Rebecca Earnshaw from the Department of Surgery, and Ella Binzing Song from the Department of Pharmacology and Toxicology. Take it away, guys.
Hello, and thank you for the introduction. So I'm Jinghu Gao from the McCraven Lab at the University of Toronto. So today I'm going to talk about my project on identifying probiotics that modulate mitophagy in models of mitochondrial dysfunction. So I'll start with some background information of this project. Pink-1 and parkin are two essential proteins that are involved in the pink-1 parkin-dependent mitophagy pathway, which is a mitochondrial quality control pathway that removes damaged mitochondria from the cell. And mutations of genes encoding pink-1 and parkin that disrupt this process are commonly found in early onset familial Parkinsonism because the accumulation of damaged mitochondria and mitochondrial oxidative byproducts in cells could contribute to the development of to the loss of dopaminergic neurons in Parkinson's disease, and there's currently no cure for that. What is interesting is that although mutations in pink-1 and parkin encoding genes in humans oftentimes lead to the development of Parkinson's disease, pink-1 knockout mice are in general healthy and show little to no PD-like symptoms. However, a study done in 2019 have demonstrated that intestinal infection with gram-negative bacteria in pink-1 knockout mice led to PD-like symptoms in those mice and loss of dopaminergic neurons. And this study suggests that there is a potential connection between the health of the GI tract and the health of the central nervous system. And this is supported by the existence of the gut-brain axis, which is a bidirectional pathway allowing the communication between the GI tract and the central nervous system. Further on, there was a high comorbidity found between the neurodegenerative diseases and GI disturbances, such as diabetes, diarrhea, constipation, and gut dysbiosis. So one commonly used method to restore the health of the GI tract is by administering probiotics. And previous preclinical and clinical studies have demonstrated that some probiotics have neuroprotective effects on patients with Parkinson's disease or Alzheimer's disease. However, the mechanism underlying these probiotics' neuroprotective effects remains unknown, which led to our question of could some probiotics exert these neuroprotective effects through a mechanism involving the modulation of pink-1 parkin-dependent mitophagy. And to answer this question, we decided to utilize both biochemistry and nutritional science approaches, trying to identify first trying to identify those probiotics that modulate pink-1 parkin-dependent mitophagy, and then to determine the molecular mechanism underlying those modulating effects. Moving on, we'll also need to determine whether those probiotics that we identified have neuroprotective effects as a food supplementation in Drosophila Parkinson's disease models. And so the first step of our study is try to identify those probiotics that could modulate mitophagy under mitochondrial stress in a cellular level. And this will allow us to narrow down our target for further in vivo studies. So previously, Peter, who is a postdoc in our lab, have identified 5 probiotic strains that had the ability to modulate mitochondrial parkin recruitment, which is an early step of the pink-1 parkin-dependent mitophagy under mitochondrial stress, and the name of the probiotics are listed here, which laid the foundation for my next step, which is to determine whether these probiotics he had identified could also modulate other steps of pink-1 parkin-dependent mitophagy, such as the serine 65 phosphobutyrinization under mitochondrial stress. And to do that, I have utilized a cellular assay with the HEC2NET3 parkin GFP cells. And as the name implied, these are HEC2NET3 cells that stably expressing a GFP-tagged parkin. And when mitochondria are healthy, parkin, which is a cytosolic protein, will not be recruited to the mitochondria and thus will distribute in the cytosol. And this can be visualized as a evenly distributed GFP signals in these HEC2-NET3 cells as represented by the schematic here. However, if I treat these cells with CCCP, which is a patornivore, that can disrupt mitochondrial membrane potential, PINK1 can then will then accumulate on the outer mitochondrial membrane of these damaged mitochondria, phosphorylating the basal ubiquitin on the outer mitochondrial membrane proteins. This process activates and recruits parkin to the outer mitochondrial membrane, and parkin as a E3 ubiquitin ligase will then conjugate more ubiquitin onto the outer mitochondrial membrane proteins, and this ubiquitin again will be phosphorylated by serine 65 by PINK1. So if I stain these cells with antibodies against serine 65 phosphoubiquitin, and then tag that with a red secondary antibody, then this red signal against the serine 65 phosphoubiquitin should co-localize with the GFP parkin signal because they both localize to the outer mitochondrial membrane, forming this yellow focus around the nucleus as demonstrated in the schematic here and in the zoomed in image here. So for this assay, testing the probiotics effect on modulating serine 65 phosphoubiquitinization of damaged mitochondria, I have established a time and concentration of CCCP treatment so that under only CCCP treatment, only about 10% of the HEC2-NET3 parkin GFP cells show this recruitment of PINK1 of parkin and serine 65 phosphoubiquitin. And if the probiotics have the ability to enhance the serine 65 phosphoubiquitinization of damaged HEC2-NET3 parkin GFP cells under mitochondrial stress, we should, after we added the probiotics to the HEC2-NET3 parkin GFP cells and then treated them with CCP, we should see a higher percentage of cells expressing this yellow foci around the nucleus. And that was exactly what we have seen here as shown in the image here. And one thing to note is that if we only treat the HEC2-NET3 parkin GFP cells with the probiotics and without the CCCP treatment, that did not induce any parkin nor serine 65 phosphoubiquitinization of the mitochondria, and suggesting that probiotics alone do not trigger mitophagy in these HEC2-NET3 parkin GFP cells. And from the quantified data here showing the graph, you can see that 4 out of the 5 probiotic strains or blend identified from the parkin recruitment assay also had the ability to enhance the serine 65 phosphoubiquitinization on mitochondria at different concentrations. So up to this point, we have identified the probiotic, 5 probiotic strains or blend that enhance parkin recruitment under mitochondrial stress out of the 49 probiotic samples provided to us, and demonstrated that some of those 5 probiotic strains or blend could also modulate other early steps of parkin-dependent mitophagy, including serine 65 phosphoubiquitinization and the degradation of mitofusin 2, which is a mitochondrial fusion protein, and that data is not shown here. So moving on, the next question is to determine what is the active components that enable these probiotics to modulate mitophagy, and also simultaneously move on to determine if these 2 probiotic strains we have identified have the ability to improve the impact of motor function in the models of a chemically induced Parkinson model of Drosophila. So our project here focused on the exploration of the basic biochemistry underneath the probiotics effect on mitophagy and how that could lead to a potential neuroprotective effects of those probiotics in Parkinson's disease. And I think it is important for patients and their family to gain access to this kind of information. And we can achieve that by attending patient interacting seminars or compose a lay language summarization of this project after the paper is published and put that summary onto websites such as the media section of the website of our collaborator Lallemand company. And at the end, I would like to thank everyone from my Cuban lab, my committee member, and Dr. Tomskin from the Lallemand company, and all the funding agencies to this project. And special thanks for MITO2i for giving me this opportunity to talk. And that's it. Thank you for your attention.